Hi. Hi. Okay, so I'm going to begin my presentation now. I guess I'm going to talk about genome analysis or human genome analysis, and I'm going to talk about understanding and annotating promoters with statistical models. And just 1 second, a little bit about me. So I'm a bioinformatician. I do computational analyses, and I'm at Yale University. And if people want, they can download the slides from the link on the first slide. So now I'm going to go to the second slide, where I will talk about how we think about genome annotation. And the way I like to think about how we might annotate the genome is to sort of think about how we, where the word annotation comes from, and how we annotate other things besides genomes, in particular text. So annotate really refers to creating kind of footnotes and side notes about a piece of text that describes the function of that text, what it's doing, and provides additional information about it. And so I extracted a little bit of text here to kind of show you what I mean, and this is actually selected on computer programming, something that I like to do, from this magazine called American Scientist. And so you see the beginning of an article, and you can see in the article what I've done is I've kind of called out bits of the text with color. So for instance, the title is colored, the first paragraph there's a quote, block quote, and so forth. And the color describes the function of the text. Obviously, the different pieces of the text mean different things. So there's one piece of text, for instance, is describes what this is about, another describes who wrote it, another introduces it, another is kind of supporting illustration, and so forth. And so, you know, one bit of annotating is kind of almost sort of highlighting things with different colors and essentially creating kind of functional annotation for it. Now, another type of thing people do when they annotate text is they look for how similar a given text is to another text, or how similar it is within a particular text. And so, for instance, in this text, the word programmer is repeated many times, and I've kind of highlighted that, and it's a kind of repeated word or leitmotif. And you know, often might find a bit in one text that's directly quoted or directly refers to another bit of text. And so often textual annotators will highlight these bits and put footnotes or some sort of reference. And that's another type of text, and that's really a type of annotation through looking at textual similarity. So now we're going to come to the process of genome annotation, and we really want to think of it the same way. It's the same type of process. And so on one hand, we can annotate genomes through comparative genomics, taking the human genome and comparing it to itself, or taking the human genome and comparing it to related humans, or even to a more distant organism such as the mouse. And when we make these comparisons, we find various blocks that are very similar between things that are very conserved, or blocks that are repeated at different scales, and so forth. And those form one type of annotation. The other type of annotation, of course, is a functional annotation. And here what happens is someone does a high-throughput experiment. They used to be, for instance, say, array-based experiments. Now everything is next-generation sequencing, where one gets a readout across the entire whole genome of kind of a functional signal. And usually this readout is a very noisy signal that the sort of computer analyst will take and smooth it, and then they'll essentially peak call it or threshold it to find various annotation blocks. And these annotation blocks then, of course, relate to what that underlying signal meant. And for instance, the signal might describe how bits of the genome are transcribed under certain conditions, or they might describe how various bits of the genome are bound to, or how they're modified. And you know, so those functional signals and functional blocks from them become a form of genome annotation. So those are the two basic forms. And then the next thing I'll say about genome annotation is that just how this is carried out. And you know, the human genome and genomics in general is fairly well known for being a very big data discipline, but also a big people discipline. This has really historically been carried out by large groups of people working together. on things, and this is really related to the original human genome sequencing around the dawn of the millennium, around 2000. And then when people came to annotating the genome, they created a sort of big group of annotators. This is the well-known ENCODE consortium, of which I'm part of, that's really worked on annotating the genome. So I'm going to talk to you about products of that consortium today. And the consortium had some initial pilot projects of, ending in 2007 on about 1% of the genome, and then 2010 on some model organism genomes. And then in the last few years, it's had a kind of more production rollout on the whole human genome, and most recently a kind of comparative rollout, comparing an updated version of the human genome to the genomes of worm and fly. So I'm going to tell you about some results kind of from that. So first of all, I want to tell you about the data corpus that I'm going to be looking at. And I'm not really going to talk at all about the tremendous amount of effort that went into generating this data, or exactly how the data is generated, because it's really quite a substantive effort that I'm not going to give justice to in that short amount of time I'm going to present. But I'm going to basically say it's a really large amount of data that's been collected in a very organized and uniform way, so that one can really slice through it and analyze it very systematically. And so the data set basically is a data set looking at many cell lines associated with the human genome, mostly looking at human adults, but also looking at embryonic stem cells. And there's a lot of data collected both on the transcriptome, the readout of the genome, but also regulomes, what factors are binding through ChIP-seq experiments, and also how the sort of histones are modified in various chromatin experiments, and I'm going to talk about that. And then there's also kind of matching, or as best match as possible, data sets for these two model organisms, worm and fly. And these data sets are, some of them are collected on cell lines, but most importantly, they're also collected on the tissue of the organism and through its time course of development. So one can watch the development of the worm and fly from an embryo all the way to an adult. And in total, the data set is very large. The whole thing I'm talking about, it's well over 100 billion reads, and comprising more than 1,000 different replicated data sets. And just looking at the RNA-seq section, which I focused on a bit, that in itself has about 65 billion reads, and a little shy of 600 distinct data sets. So now I'm moving on to slide 6, and I want to sort of talk to you a little bit about the kind of goals of the kind of comparative part of this analysis, and how one wants to do a kind of functional analysis and a comparative analysis at the same time. So when one thinks about doing a comparative analysis, one of course thinks about how related the different organisms are. There are things that are fairly close, like human and mouse, things that are much more distant, like human, worms, and flies, and so forth. And so people in the past have compared fairly closely related organisms, say human and mouse, in terms of their, say, transcriptomic data, or in terms of one functional genomics data set. Or moving on to the next slide, they've looked at kind of more integrated types of things, putting many different types of functional data together for an individual organism. Now, this, of course, was previously done by ENCODE for human, also fly and worm. People have done this for yeast independently. They've also done it for mouse. And what I'm going to endeavor to talk a little bit about today is kind of to do both like very distant comparisons between, say, human, worm, and fly, but also to do those comparisons in an integrated way, looking at many of the different types of functional readouts, the transcriptome, the sort of regulomes, looking at the binding data and the readout data. So now moving on to slide 9, the first thing that people have looked at here is how different the genes are in these different genomes, and they've compared the genes in these things. And this picture shows just the sort of how many genes are in the different organisms over time for like the past 10 years. And the important thing you see in the picture is very flat lines. And so the annotation of the genes in the genome has become fairly stable. Now, there's still a lot of work to be done in terms of getting their exact splicing structure and so forth worked out. But the overall number of genes in the human genome has been fairly steady. The number in the worm genome has been fairly steady, and the number in the fly genome is fairly steady. for the last 10 years. And, when people look at the gene statistics, on a very aggregate level, they see, a lot of the genes in these organisms do look fairly similar. The size of the exons are fairly similar when you look at the distributions. The longest open reading frame per gene is fairly similar. Human genes tend to be a little bit longer than in total gene span than the other ones, and that's often because of the number of exons per gene, and also because of course, the size of their introns. So, of course, the really interesting thing when looking at the comparative genomics and also the functional genomics is not so much really looking at the genes in the genome, but looking at the non-coding regions of the genome, where people really know less about them. And there's really, in a sense, much more to do. And I like to kind of introduce these non-coding regions by sort of talking about them as the dark matter of the genome, and making a comparison to the dark matter that we see in general in the universe. So the picture, the slide shows an actual picture of dark matter. So that's a picture through a telescope of dark matter. And what you see when you look at dark matter is you don't see it. You see just the stars. But the way astronomers see the dark matter is they see it by the way it affects the light coming from the stars. It essentially lenses that starlight, or regulates it, bends it. And that's, I think, a very good analogy to the dark matter of the genome. A lot of the dark matter of the genome, what it does is it modulates the effect of the genes. It's involved in the regulation of the genes when they go on and off, how much they're transcribed, how they work, and so forth. And so I think that's the kind of picture we're going to be talking about today. We're going to talk a lot about gene regulation. I think dark matter of the genome is also important, because a lot of the disease association people are finding more and more is really in relation to these non-coding regions of the genome. So moving on to slide 12, what I'm going to talk about today is focusing on one particular part of the dark matter of the genome, and that's really the part that sits right upstream of the gene, and that's the promoter of the gene. And one of the key aspects of these promoters is are they active? And the activity of the promoter is often described by kind of how open it is. And so you can have the DNA upstream of the gene is often wound around these histones. And when the gene becomes very active, it becomes kind of very open, and when it becomes very repressed, it's very closed down. So some key annotation questions that we are going to sort of talk about are, how do we define these, where the actual promoter annotation is? And then when we have an active promoter, how do we talk about its degree of activity in terms of how many transcription factors are bound, or what the chromatin looks around it, or its epigenetic marks, and how we relate this to transcription. And then we want to talk about how conserved these kind of ideas and principles are amongst different organisms. So are they very similar, or are they very different? Do they represent very fundamental things or very unit-specific things? So moving on to slide 13, the answers to these questions that I'm going to talk about, I think they're kind of significant because on one hand, they sort of tell us about kind of very conserved and ancient principles of gene expression in animals. Of course, they're also very useful in helping us better annotate and understand what each base in the genome is actually doing. And then in particular, in relation to sort of epigenetic changes and so forth, I think they're very important in some diseases, because, for instance, in cancer, we often see a lot of global epigenetic changes in the genome, and what we really want to know is how those changes are coupled to new genes being turned on or turned off and so forth. And so what we're going to talk about today is going to be related to that. So now moving on to slide 14, what I'm going to talk about is sort of shown in this little mini outline. So the first thing I talked about was kind of a little introduction to genome annotation and the ENCODE and the data we're going to look at. And so then I'm going to focus on two things in the rest of the talk. I'm going to first talk about how we relate the histone marks upstream of the gene to the gene expression, and how it works, kind of how to develop a kind of a very general model for this. And then I'm going to talk a little bit about how we relate the transcription factors that are binding upstream the gene to its expression, compare them to the histone marks. And one of the surprising things we're going to find is that very few transcription factors, I think, are actually, surprisingly, very few transcription factors can give very successful prediction of the output of a gene. And so those are the two topics that we're going to cover in detail. So now going to slide 15, let me give you just a little schematic of the problem, the way we're going to pose the problem. So the picture shows two sort of horizontal dark lines, and they represent the genome in two different cell types. It's two different bits of activity. And you can see there are genes on the genome, and the level of expression of the genes is kind of explained by the little green things, you know, how on and off certain genes are. And then the promoter directly upstream is a little blue line that, and the activity of the promoter in terms of how many things are bound to it, or how it's how much the degree of its histone modifications is indicated by the amount of the red things. And so what we want to basically do is we want to make a plot where we plot the amount of the red lines for each gene to the number of green lines. And so we would do that over, say, the 20,000 genes in the human genome, and then we'd have a plot such as the one shown on the bottom, where we relate expression to some measure of promoter activity. And, you know, ideally we'd want them to be correlated so that we can almost predict or relate the promoter activity to the expression level. And now we can do this actually for more than one cell line, because I think one of the key ideas is that in different types of cells, because of the different epigenetics, the certain promoters are going to become active and others not, and we want to look at the kind of changing epigenetics and think about how that is going to be related to the changing gene expression. And so that's the overall cartoon picture, and that little XY plot shows the kind of cartoon. So let me start to show what it looks like with real data. So on slide 16, I showed just a very basic picture where if we just look at, say, one histone mark, that's H3K4ME3, and another thing that's the binding of a factor that's very associated with transcription polymerase, what does it actually look like? So on I'm showing this actually for data from the worm. This is just a representative plot. And on the y-axis, you see the kind of input, you know, the histone mark or the polymerase binding, and on the x-axis you see the output, the expression. And you can see a kind of rough correlation. And let's just focus on the numbers. Roughly you see about a 0.6 sort of Spearman number for both of those correlations. You know, so each of those sort of inputs are roughly correlated with the output, but not perfectly correlated. I mean, 0.6 isn't perfectly correlated. And so what we want to do is see if we can do better and see if that gives us some further understanding. And so that's what I'm going to talk about in slide 17. We tried to make a more sophisticated mathematical model of this process. So what we did is we looked around the TSS, the start of transcription of the gene, and we made little bins right up in front of it and right after it, and also the same at the end of the gene. And then in each of these bins, we put in each of them the amount of the particular chromatin features, the histone modifications, or as I'll tell you later, we did the same thing for transcription factors. And then we also put the correlation of this one modification with the gene expression level across all the genes. Okay, so now we have all these different bins with their different correlations, and now what we're going to try to do is relate those correlations to the gene expression level of the gene from the RNA-seq. That's the overall set of the experiment. And then in slide 18, the next slide, you can start to see a little bit of how the data looks. So now looking at the right-hand side of the slide, you see this sort of heat map where we have many different histone marks, and this is a very simple heat map for a model organism. This is for worm, where you see for each of the bins the degree to which that bin has a correlation with gene expression. And you can see that the red represents more correlation, blue represents sort of anti-correlation. So you can see some marks like H3K9, ME2 are repressive marks, they're anti-correlated. Some marks like H3K4, ME2 are red, they're positively correlated, and the correlation really depends on the position. You know, sometimes the correlation is much stronger close to TSS which weaker farther from the TSS. So now what we're going to do is put all of these things together into a mathematical model, and that's what's shown on slide 19. And so we're going to put them together into two mathematical models. So first mathematical model we're going to make is just simply a classification model, and we're going to use this particular type of classifier called to support vector machine. I'm not going to describe it in detail, but basically it puts a lot of the different the correlations from a lot of the different marks together. And what we try to do is classify genes to highly expressed versus lowly expressed. And now the first thing we can do is we can see how we do, and that's shown at this what's called the AUC curve, which is shown at the top left hand of the slide. And we can make this AUC curve for all the different bins upstream of the TSS. And you can see some bins get a better AUC. There's more area above the curve, and some a lesser AUC. And so we can plot then on the bottom left-hand figure the overall score for each of the bins. And so we can see which of the bins in front of the TSS is most predictive. And as you might expect, the bins right around the TSS contribute most to the prediction, and whereas those more distant contribute less. So then the next thing we can do is we can move from doing a classification model to doing what's called a regression model, where we try to directly predict the overall level of expression of the gene through putting together all the different marks. And so I show that on the top right-hand side. And so you can see this is for a model organism. You have the observed expression versus the predicted expression for 13 different histone marks, integrating them now with this mathematical technique called support vector regression. And you can see we get a pretty good line. There's a good R here. It's about an R of about 0.75. This is pure sin correlation. And then in addition, what we can do is we can take our model that we've parameterized, trained and tested on protein coding genes, and we can apply that model without modification to the promoters of non-coding RNAs, particular microRNAs, and actually we get some correlation there, which is also quite impressive since we never really parameterized the model against them, but it's being used to predict on the basis of them. Okay, so now let's go to the next slide, slide 20. So now what we can do is we can we have the epigenetics and we have the RNA-seq readout for many different types of cells. And for instance, in human, we'll have many cell lines. In the worm and the fly, we'll have many different developmental stages. And so we can play this game where we say, oh, well, how well does the epigenetics in, say, one particular stage predict the expression in that stage or the expression in another stage? Now, naively, you'd expect, and correctly, that if you match the stages, you would get the best prediction. And lo and behold, that's what we see. So, for instance, these two the classification curves in the graph show the prediction of the embryo gene expression level from the embryo epigenetics, which is the blue line, and then the other lines are shown based on other stages, the larval stages, the epigenetics in those, and you see you do a lot worse. And so this, of course, shows that it gives you a measure of how the matched the epigenetics are to the actual readout and expression you get in a particular stage. Okay, now moving on to slide 21. So now I'm going to move from model organism, which is more of a simple setup, to human, and I'm going to show you the results. This is from the ENCODE consortium paper a while ago. The sort of best we could do on human, and this was it's a little complicated how this thing was set up with both a classification and a regression model. But the bottom line is when you put all the points there for human, all the genes down, you can do really well. The measured expression is shown on the Y, and the predicted expression is shown on the X. And the overall R you get is about 0.9, which is a very, very good prediction. And especially as far as bioinformatic predictions go, this is quite a good prediction. And in addition, you get for each, in the model, you get a sense of how important each of the individual histone marks are in the model. And that's what's shown in the bar graph on the bottom left. And I'm going to come back to the relative importance of these histone marks in a second. So then the next thing we can do is we can do the same thing with the same model and kind of compare many different organisms. And so here I show just trying to build a model for worm, trying to build it for fly, trying to build it for human, build it for yeast, build it for mouse. And the bottom line is you can do relatively well in all the organisms if you build a model. You can see they look slightly different. The R's are slightly different. But overall, you seem to be able to do this kind of predict expression from the histone marks fairly consistently in all the in human and many model organisms. And so that brings us to the idea of maybe we can actually compare the models that we build and even go farther and try to build some sort of universal model that will work across many organisms that really represents some basic and ancient features of transcription. And so that's what I'm trying to do in slide 23. So here what you see is you see those little pictures that describe the correlations with gene expression for all the different histone marks upstream of the TSS for 3 different organisms, human, fly, and worm. And the human is kind of reddish, the worm is kind of greenish, and the fly is bluish. And you can see the patterning is fairly similar, but it's not exactly the same for the human, fly, and worm, for all the different marks, but not exactly the same. And then what we can do is we can build a model for each of them, and then we can look at the relative importance of each of the marks in that model. So for instance, the importance of the marks in human are shown by that very big red bar to the top right, and then the importance of the marks for worm are shown by the green bar and fly the blue bar. And what you can see is the different marks have different importance in the model. For instance, in human, H3K4ME3 is actually quite important, whereas in worm and fly it's less important, and so forth. And that seems to vary. And that maybe reflects in the fact that in human, there's really a different scale to the upstream region than in worm and fly. And so then what we can do is we can actually imagine, if we take the structure we have for the models, we can imagine to try to combine the models and sort of simultaneously build a model that would work without change on all 3 organisms, human, worm, and fly. We call this a universal model. And the importance of each of the marks in that universal model is shown by the black bar in the right-hand side figure with the word universal over it. And so you can see in that particular model, H3K4ME2 and H3K4ME3, which are very well-known promoter marks, are the most predictive or the most important marks in the model. And something like let's see, the H3K27 acetylation becomes less important, whereas in, say, the human model, it was more important. And so it's interesting to see these differences in which marks are kind of the most ancient and the most Have I dropped off my online? I hope I'm still online. My screen, yeah, okay, thank you. My screen just blanked for a second, and I was very worried that I had dropped off. In any case, we're on slide 23, and I was talking about the relative importance in the different organisms of the different marks and talking about this universal model. And then the neat thing is we can take the universal model and we can see how well that model does in predicting relative to organism-specific models. And so that's what we did. And the grid on the top right-hand side, we look at developing a model, say, in human and predicting on human, developing a model in, say, human and applying that model to worm, developing a model in human, applying it to fly. And in those contexts, you can see the R that you get. And the human, obviously, if you build a model in human, you predict on human, you're going to do much better than if you try to transfer that model directly to worm and fly. And likewise, you build a model on worm, you'll do better, and so forth. But what's really neat is if you look at the bottom part of the right-hand thing, you can see the prediction accuracy of this universal model. And so the universal model does almost exactly well as the organism-specific model. So for instance, in human, an organism-specific model gets an R of 0.82. whereas the universal model will get a 0.8. And in fly, it will get 0.84. The universal model will get 0.83. And so that's really quite impressive. It says I can develop a set of parameters for a model that work equally well on human, worm, and fly as developing a model in that organism specifically. And it says that we can really talk about the relationship between the upstream chromatin structure and the level of gene expression in a very basic and fundamental way. And then the other thing we can do, which is particularly neat, is we can take our model, as we did for the organism-specific models, and we can apply that model, which is parameterized across all the organisms on protein coding genes, but now we can apply it to non-coding RNAs. And lo and behold, the model actually is very successful. For instance, in human, it gets almost 0.7 accuracy in predicting non-coding RNAs. In worm, it doesn't do as well, but I think that's because of the fairly poor TSS definitions in that organism. Okay, so now we've finished talking about the histone mark models, and I'm going to move on to the TF model. So I'm moving through my outline slide to now slide 26. And so now we're going to do the same game that we just did for histone marks, but we're going to do it for transcription factors. And so for instance, at the top, you see I'm just showing a representative picture where I have a number of worm transcription factors. I have bins upstream of the TSS, as I did for histone marks, and I'm correlating the binding of those things with the gene expression. Red is high, blue is low. The neat thing you can see here is there's a real kind of bipartite structure to the figure. There's activators that are highly correlated with gene expression, and then there are repressors that are negatively correlated with gene expression. And likewise, there's some factors such as ELT3, where they seem to have all their correlation right upstream of the promoter, and other factors such as CEH14, which seem to be more like histone marks, where they seem to be to sort of have activity over the entire promoter. And then more directly in the bottom part of the figure, I then compare the TFs between different organisms, between the human, which is the red shade, the worm, and the fly, just looking at the different patterns. So then in slide 27, we do the same thing. We try to build a model to predict the gene expression. Now this is a little bit more complicated model. It's a sort of a multi-layer model where first what we do is we integrate all the information, the correlations in one bin, using the support vector regression for, say, one bin. We do that for all the bins upstream of the TSS. And then we have another layer of our model that takes the output of all those support vector regressions and puts that into one model where we make a combined prediction. And then we compare that to the gene expression level, or to the RNA-seq. And how well do we do? So here's the results we have for human. Now remember, for human, we could get about 0.9, the best, that's what we could get with the joint classification regression model. Here we get 0.81, which is good, but it's not as good, but it's pretty good, and so that's using the predictions of the TFs. And of course, here what we can do is rank many of the TFs by their relative importance in the model, and we can see some TFs, for instance, YY1 has a greater predictive power than others. The other thing we can do is, in addition to predicting the gene expression of a gene, we can also predict its differential expression. If we have the gene expression in two different cell lines, we can look at the difference in expression, and if we have the epigenetics in those same two cell lines, we can look at the difference, the change, the delta epigenetics between the two cell lines, or the differential TF binding between the two cell lines. And we can find that the differential TF binding, how much the TF binding changes, is actually fairly predictive of the gene expression. Not as predictive as the absolute, but fairly predictive, and that's what's shown in the heat map in slide 29. And then in slide 30, the other thing we can do is we can look at the relative importance of the TFs in different cell lines. For instance, one cell line is K562, another is GM12878. And what's interesting is that some TFs are more important in some cell lines than others. So for instance, in K562, YY1 is particularly important, and SP1 is very important. In other cell lines, different TFs become very important. For instance, REST and ELF1 are very important in GM12878. And that's sort of associated with the transcription factors that we know to be very important in those particular lineages. For instance, ELF1 we know to be very important in the white blood cell lineage. Then the other thing we can do on slide 31 is we can compare the histone mark models and the transcription factor model, the TF models. And here what we can see is we can look at the relative importance of each of the bins upstream to the TSS, and you can see that the TF model on the top right-hand side, it's much more peaked. The region right around the TSS is much more important in that model than the histone mark model, where it's more diffuse, and also the area around over the gene body is very important. So you can see the sort of differences between the TFs and the histone marks. And then the other thing you can do in these pictures is you could say, well, we have a TF model and we have a histone mark model. Why don't we combine them and make a super or uber model that has all of the things, and maybe we'll be able to do even better. And so we did that, and that's shown in the black line in the bottom right-hand side of the figure. And that, now, interesting enough, that black line doesn't do any better than the histone mark model. So what that implies is there's no extra information in the TF binding that's not encoded in the histone modifications. It's insofar as prediction is the case, which is kind of an interesting thing. So all of the information about the gene expression that we can get lies within those histone modifications. There's no extra information that we can get from the transcription factor binding. Not completely intuitive, but it is the case. The other thing we can do is we can look at the sort of peakedness of the TF model versus the histone model in different organisms, and here we compare that in human worm and fly, the red, green, and blue figures in relation to genes, non-coding RNAs, and you know what we basically see is that same pattern is observed across all the different organisms as we observed in a particular organism. And then the final thing we can do, which is kind of neat, and on slide 33, is we can say, well, how many transcription factors do we need to make a successful prediction? So we can take, let's say we take, of the almost 1,000 transcription factors in human, we take 5. We randomly pick 5. And now experiments weren't done on 1,000. Experiments are only done on maybe up to 200 now. We pick a subset of 5. And we can take many different subsets of 5. And we can see how well we do. And then we can do subsets of 6, subsets of 7, and so forth. And then we can, for any given number, for instance 5, we can average the results we'll get from all the different picks. And that's what you see on the red curve. And what it shows, which is quite remarkable, is that one can actually get a very good predictive performance, almost as good as a full model with all the available TFs, with only a small set of TFs. So if one only uses 5 transcription factors, just a random selection of 5, one gets 90% of the predictive power of a model with 30 or 40 TFs. And that's not completely intuitive. You might say, well, there's 1,000 TFs, each of them has great specificity for sort of gene expression. Why is it that we're so successful predicting with only a small number of transcription factors? Where's the specificity? Where's the information in the other transcription factors? And when we first presented this result years ago, people found it very paradoxical that it could be the case. But I can rationalize this, actually. I can give you a biological rationalization. I can also say that we've repeated this type of thing in many different contexts in many different organisms. For instance, in worm and fly, you get a very similar pattern shown by the blue and the green lines. Now, the biological rationalization for this is that we're measuring here steady-state levels of gene expression after the chromatin has been opened. And one imagines after the chromatin has been opened, one has some transcription factors that are bound, maybe opportunistically, maybe not even specifically, that are more a proxy for how much the chromatin has been opened. So the more the chromatin is opened, you get more kind of low-level opportunistic binding of the transcription factors. And this opportunistic binding doesn't reflect as much this specificity of actually opening up the chromatin dynamically and setting up the initial gene expression. This being done by some pioneer transcription factors that come in, open up the chromatin. That's not at all shown by these correlations, and what you're seeing here is more the sort of steady state level of gene expression and how the TFs really are just mirroring back the results of the chromatin being opened. The other thing we can do with the transcription factors, I'll briefly just say, is we can take them and we can arrange them into a transcription factor network. I'm not going to describe this in detail. This is described in other works. And it turns out in this network we can build a bit of a hierarchy where some other factors sit at higher levels than others. And I won't describe how this is built, but other than to show you a selection of roughly 120 of the human transcription factors create this three-level hierarchy that you see in the top left-hand side of the figure. What's kind of neat is we can look at each of the factors in the hierarchy and calculate its overall power in the model in terms of driving gene expression. And we can color each of the factors by its overall power. And that's shown by the blue tinge to each of the factors in slide 35. And what you can see is kind of neat is that the factors on the higher levels of this hierarchy tend to have slightly stronger correlation or slightly stronger power in the model to drive gene expression. Sort of the idea being that the more higher up you sit in the hierarchy, the more influential you are in terms of driving gene expression. So now moving to slide 36, I'd just like to summarize my talk. So today I talked to you about something to do with human genome analysis. In particular, I talked to you a lot about really looking at promoters with statistical models. And so first I introduced how we do the genome annotation. In particular, I introduced the large corpus of data from the ENCODE project that we looked at, the large amount of human data, also data on model organisms. And then I focused on kind of two slices of the data, connecting essentially the inputs, the histone modifications and the transcription factors, to the outputs, the RNA-seq. So first we built a histone model of gene expression. And some of the key things from a model is it works pretty well, and it actually works for non-coding as well as genes. It's fairly tissue-specific. And what I think is kind of neat and not completely intuitive is we can actually build this universal model, one set of parameters that works on everything, works on protein-coding genes, non-coding RNAs, works across organisms, and so forth. And then I talked about building transcription factor models, and I think the key take-homes from this are that you can see here that you get the different importance of the different bins in front of the TSS in terms of the importance for the model. And here you can see how the TFs are, the region right in front of the TSS is much more important for the transcription factors than the histone modifications. And this is very important in terms of defining the actual promoter of the real important regulatory region. And then also you find, though, that in terms of the statistical models, the signals we get from transcription factor models and histone mark models are very redundant. So there's no additional information in the TF models. They're really just mirroring back the information in the open chromatin and histone mark models. And surprisingly, because of this, we get the surprising conclusion that actually only a small number of TFs are quite predictive of gene expression. You can achieve the predictive power of a full model with only about 5 transcription factors, which is quite not intuitive, but I tried to rationalize biologically. And then in slide 37 and 38, I'm going to acknowledge things. Like I said, this is all this work is part of this bigger ENCODE and modENCODE consortium. There's lots of people involved, and in this particular slide here, 37, I show all the people involved in what's called the modENCODE transcriptome group. These are all the people that really focused on the transcription in which we built these models. And I didn't even talk in the presentation much about the large-scale data generation, but a lot of the people in this group, of course, are generating all these huge and wonderful data sets. And in particular, we need to acknowledge Bob Watterson, Tom Gingeris, and Sue Selnicker, who led the data generation efforts on worm, human, and fly, respectively, and also Brett and Gravely participated in this very much. And then the other people listed on this figure are a lot of the analysts, and I kind of highlighted some of the key analysts who worked on this, Joe Rosowski, Chao Cheng, Kun Q, Yan, and so forth. Now, the person who really was the key analyst for the specific stuff that I talked about today is Chao Cheng. And so if we move to slide 38, I actually have more specific acknowledgements for Chao because he developed a lot of the statistical machinery in separate, sort of smaller scale, separate works, and then applied it to the big consortium. And so Chao was a postdoc with me for many years. He's now moved on to a faculty position at Dartmouth, and he worked very extensively with Kun-Kyu Yan, Nitin Bharadwaj, and Ren-Kun Min, and others, and also Xiping Wang at BU in developing these models. And with that, I'd like to end. I'd like to, in a final note, also say that I'm keen to recruit additional new postdocs, talented people like Chao, for my lab, and there's a URL for that on the slide. Thank you very much for your attention. So I'm all done. Oh, there are questions. I see. Okay, I click on. Sorry, I'm learning about this. I click on the Q&A here. Okay, so I see now. So the first question is, how might we use a human text to treat cancer? Well, that's a good question. I mean, that's how I introduced the talk, and I think that it's obviously we're not going to literally do that, but I think a lot of the machinery that we developed to annotate human text and the approaches that we use to do text mining and the ideas we have, there's some transferability to how we can use that to annotate the genome. And annotating the genome, I think, is very important in terms of treating cancer, because cancer is a disease of a truly messed up genome. You know, our genome gets really messed up. We have cancer, it gets rearranged, the epigenetic changes change very much. So I think the one can transfer a lot of the mathematical ideas, and I think that's quite useful. Now let's see. So the next question was, how do we control the active regions promoters? Well, I talked a little bit about that in the talk. You know, really when how the promoter becomes active when the chromatin's open from the histone modifications, and then I talked a bit about also the transcription factors binding and what activity we see in the promoter regions and how we relate that to gene expression. So now moving on to the next question, see direct relationship to rocket ship model. Well, this was a bizarre question, so I think we're going to leave this one. So then the next one was, have you formed these models with cell-specific data? If yes, what do you mean? Can you see more information coming from specific histone marks or from TFs as a group? So yes, now all these models are based on cell-specific data. So one takes a given type of cell or a cell line, and one collects an RNA-seq data set and many different ChIP-seq data sets. The ChIP-seq data sets coming from transcription factors that are binding in that cell line, or histone modifications in that particular cell line. And they have to be very cell line matched, because I think, as I showed you early in the presentation, if we take the histone modifications or transcription factors from a different cell line, they're much less predictive of gene expression than in the matched cell line. And that makes a lot of sense, because that's really how the epigenetics exerts its effect by it changes, and that turns the genes on and off and so forth. Then there's another question, number 6 here. Can the data generated from these studies be used to assess active promoter regions for targeted genes and possibly correlate with the effect of SNPs in those active regions? Yes, most assuredly. I didn't talk about that, but that's what we're quite actively working on. And so the whole point is, if you can use these bins to figure out which of the regions of the promoters are really important, and where the epigenetic changes in the transcription factor binding really affects things, you obviously know where the regions that are more sensitive to the sequence variations, the SNPs and the structural variants are, eventually we hope to be able to really understand if a given SNP, or even more to the point, a somatic alteration that you might have in cancer, will exert effect on transcription in the promoter. Now let's see. So, question 7 was, how many features were selected for classification and regression analysis? So that's a very good question. Now, I talked to you about a lot of different analyses we've done, frankly, over a number of years, and you know, the analyses aren't exactly matched. I mean, some we used a certain group of factors, others a different group of histone marks. But let me just give you a good sort of way to think about this is if you go back to slide, let me see if I can quickly find that slide. If we go back to slide 28, there's our human, all the human factors that we looked at, and that basically shows you we have about 25 factors in a cell line that we're building a model on, 30 factors, maybe more, maybe 50, but that's the number we're using for a lot of these models. And then, for instance, in slide 23, we show the key marks that we used that were kind of in every organism. We used maybe 7, 8 marks we used to make these histone mark models. Okay, then moving on to question 8, the question was, thanks for your talk. You have mentioned about developing cross-species model. How far will be valid for TF from various eukaryotic, prokaryotic organism, or is it only for eukaryotes? So first of all of this is only valid for eukaryotes. I mean, a lot of this, the chromatin discussion, and really all of this is really a eukaryotic discussion. We've never thought about applying it for prokaryotes. I mean, potentially for the transcription factors, some of it might be transferable. We've never done that. Now, we've not really developed universal cross-species models for the TFs, and that's because we don't have enough true ortholog data sets for the TFs. Now, for the histone marks, we have the same histone mark, like H3K4, ME3, being collected in many different organisms. For TFs, you know, we have polymerase in many organisms, but we don't have many other TFs where we have the same TF in many organisms. So that will really have to wait for further data sets in the future. So that's question number 8. And then moving on to question number 9, how did you decide which TFs bind to the promoter regions? If it's purely by binding sites, how do you eliminate the large amount of false positives since most binding sites are short? So that's a good question. What we do here, it's a little different, was we're only looking at the promoter. So first of all, we're not looking at the binding outside the promoter. We're only looking at the binding in the promoter. And we're not per se calling whether something is bound or not. We just basically have a continuous readout signal from the ChIP-seq experiment. It's very close to the data of just the level of binding. Now, the level could be very close to the noise level, right? It could be very low, very small number. It could be a medium number that maybe reflects non-specific binding that just reflects opportunistic binding to an open promoter, or it could be a very large number that reflects very strong and specific binding. All of those numerics go into the model. Okay, so the very small numbers, the medium numbers, and the large numbers go into the model. And there's less a sense of whether that whether the binding is specific and functional or non-specific and opportunistic, we don't differentiate against. But then in a sense, do we need to or do we want to? Maybe not. And that's the way I would answer that. And so that's a question from Queen Yang, and I'm done with that. And then it says, I have no questions available for display. Oh, hold it. How would you, no, there seems to be one more here. Number 10, how would you suggest to annotate SNP or other types of genetic mutation with chromatin modification data? So that's a very good question. I think the key, I don't think one would annotate the SNPs themselves with the chromatin modification data. I think a better way of doing it is to use the chromatin modification data to annotate regions in the genome and then talk about where the SNPs lie. So does the chromatin data say we lie in a promoter? Does the chromatin data say we lie in an enhancer? And then is that enhancer or promoter active in a particular cell line? And then if we have a SNP there, presumably that SNP affects a active promoter or enhancer in a given type of cell. That's the way I would approach that annotation. And again, we seem to have no questions for display. So I think that means I'm done. So let me ask our moderators. Am I done? Oh, hold it. Now it's beeping again. Hold on. So, oh, there's a question 11. What are some of the technical hurdles did you have to overcome in processing the data? So that's a very good question. Now, of course, I really glossed over big time the actual data generation and also a lot of the immediate processing of the data. So there's a serious amount of technical challenges in actually getting the data from the sequencing machines, managing the data on your disk, but then also normalizing all the data sets, so all the signals, right? Removing artifactual regions of the genome, mapping the reads correctly to the genome, dealing with mismapping or multi-mapping reads, and so forth, and getting a very pure and nice, well-normalized signal. And those signals are what then fed into my model. So this is a very downstream or derivative talk, in a sense removed from a lot of the hard and complicated work that goes with building these data sets. But of course, that's the point of building these wonderful resources like the ENCODE, modENCODE resource, where all that work is done and you can use the processed data. So I have no more questions, so I'm going to thank the audience now for listening, and I think it says I'm being turned off. So take care. I guess I say goodbye. Okay. Is that it? Am I done?